Assalamu alaikum dear students, welcome back to another video. In this video we will discuss the most important topic is DNA replication MCQs part 1st. Which phase of the cell cycle does DNA replication occur? Option A, G1 phase, B, S phase, C, G2 phase, D, M phase. Correct option is B, S phase. Which enzyme prevents the supercoiling of DNA? Option A, DNA helicase, B, DNA polymerase, C, topoisomerase, D, ligase. Correct option is C, topoisomerase. Where does DNA replication occur in the eukaryotic cell? Option A, cytoplasm, B, nucleus, C, ribosome, D, mitochondria. Correct option is B, nucleus. Subscribe now and press the bell icon. Never miss an update from early technologist. What type of replication does DNA undergo? Option A, conservative, B, semi-conservative, C, dispersive, D, non-conservative. Correct option is B, semi-conservative. Which strand is synthesized discontinuously? Option A, leading strand, B, lagging strand, C, both A and B. Correct option is B, lagging strand. What is the role of DNA ligase? Option A, synthesizes new DNA strands, B, join Okazaki fragments, C, unwinds DNA, D, adds RNA primers. Correct option is B, join Okazaki fragments. What is the direction of DNA synthesis? Option A, 3' to 5', B, 5' to 3', C, both directions, D, random. Correct option is B, 5' to 3'. Which enzyme removes RNA primers? Option A, DNA ligase, B, DNA helicase, C, DNA polymerase 1, D, RNA primase. Correct option is C, DNA polymerase 1. Which enzyme unwinds the DNA double helix? Option A, ligase, B, helicase, C, primase, D, polymerase. Correct option is B, helicase. What would happen if DNA polymerase made an error during replication? Option A, the error would always be repaired immediately. B, a mutation could occur. C, the entire cell cycle would stop. D, the DNA strand would be destroyed. Correct option is B, a mutation could occur. What is the function of DNA ligase? Option A, joins Okazaki fragments together, B, adds nucleotides to the grooving DNA strand, C, unzip the DNA double helix, D, replaces RNA primers with DNA. Correct option is A, joins Okazaki fragments together. On which strand are Okazaki fragments found? Option A, leading strand, B. Lagging strand. C. Both strands. Correct option is B. Lagging strand. Why is DNA replication faster on the leading strand? Option A. DNA polymerase works only in the 3' to 5' direction. B. The leading strand is replicated continuously. C. Okazaki fragments speed up the process. D. The lagging strand is not replicated during cell division. Correct option is B. The leading strand is replicated continuously. In which phase of the cell cycle does DNA replication occur? Option A. G1 phase. B. S phase. C. G2 phase. D. M phase. Correct option is B. S phase. Which enzyme aids new nucleotides to the growing DNA strand? Option A. DNA helicase. B. DNA polymerase. C. DNA ligase. D. RNA primase. Correct option is B. DNA polymerase. Which enzyme is responsible for unzipping the DNA double helix? Option A. DNA polymerase. B. DNA ligase. C. DNA helicase. D. RNA primase. Correct option is C. DNA helicase. Which organelle has its own DNA replication system? Option A. Golgi apparatus. B. Mitochondria. C. Ribosome. D. Endoplasmic reticulum. Correct option is B. Mitochondria. Which feature of DNA allows semi-conservative replication? Option A. Complementary base pairing. B. Hydrogen bonding. C. Phosphate backbone. Correct option is option D. Chromatin structure. Correct option is A. Complementary base pairing. What happens if DNA polymerase lacks exonuclease activity? Option A. Replication stop. B. Increase mutation rate. C. No primers are added. D. Okazaki fragments are longer. Correct option is B. Increase mutation rate. Which enzyme ensures high fidelity in the DNA replication? Option A. Helicase. B. DNA ligase. C. DNA polymerase. D. topoisomerase. Correct option is C. DNA polymerase. What provides energy for DNA polymerase activity? Option A. ATP. B. DNTP hydrolysis. C. GTP. D. RNA primers. Correct option is B. DNTP hydrolysis. Okazaki fragments are found on the option A. Leading strand. B. Lagging strand. C. Both strands. And option D. RNA template. Correct option is B. Lagging strand. What is the function of single strand binding protein SSBs? Option A. Prevent DNA strands from reannealing. B. Break hydrogen bonds. C. Remove primers. D. Join Okazaki fragments. Correct option is A. Prevent DNA strand from reannealing. Which strand requires multiple primers? Option A, leading, B, lagging, C, both strands, D, neither strand. Correct option is B, lagging strand. Which enzyme synthesizes RNA primers? Option A, DNA polymerase, B, RNA primase, C, DNA ligase, D, helicase. Correct option is B, RNA primase. Which enzyme aids nucleotides during replication? Option A, helicase, B, DNA polymerase, C, ligase, D, topoisomerase. Correct option is B, DNA polymerase. What is the role of telomerase in eukaryotic cells? Option A, proofreads DNA to correct errors, B, unzips the double helix, C, extends the end of the chromosomes to prevent shortening, D, seals Okazaki fragments. Correct option is C, extends the end of the chromosome to prevent shortening. Which base pairs with adenine and DNA replication? Option A, cytosine, B, guanine, C, thymine, D, uracil. Correct option is C, thymine. Thank you for watching my videos.